So a very good afternoon to everyone present for this session of DDH 2020 training program. We're very privileged to have with us today a very special person, Professor Indira Ghosh. Professor Indira Ghosh is a retired professor and ex-dean at School of Computational and Integrative Sciences, JNU, New Delhi. She completed her MSc in Physics from Calcutta University and obtained her PhD at ISC Bangalore under the guidance of Professor V.S.R. Rao in Molecular Biophysics, a group led by G.N. Ramchandran. She joined University of Houston, USA as Fulbright Scholar with Professor J. Andrew McCammon and contributed to novel method in computational biology. She published one of the first docking algorithms in early 80s as a part of a thesis and developed difference of free energy calculation methods in biomolecules during her postdoc. She has been amongst the earliest scientists to realize the importance of bioinformatics and initiated MSc courses in Pune University when she joined as a professor in the Institute of Bioinformatics and Biotechnology in Pune in 2003. She has been enriched with 13 years of industry and 15 years of academic experience, which led her to develop the skill of integration of analytical methods with all branches of science and technology. Her major contributions were to develop and contribute in the field of bio and chemoinformatics using system biology approach to find pathway and target enzymes and developing novel tools for molecular simulations and pharmacophore designs using known protein structure. Apart from this, she has been contributing immensely in designing courses for many Indian universities for bioinformatics and computational biology, UGC ePathshala, Bioinformatics Task Force in DBT, and European Union Commission aided multi-country projects. With a small introduction, I welcome you, ma'am, to this DDH training program, and I now hand over this session to you, ma'am.
Thank you. Thank you very much for inviting me to give this talk. And I'm thankful that you agreed to give this talk in the later part of your hackathon. I'm sure that most of the participants have already been very much influenced by the mesmerized talks done by the list of people I saw in your posters. So, and most often we all agreed upon is that designing drug is very easy. And well, it is with the help of a lot of competition tools last 50 years, it turned out to be very easy go for most of the people when we know which is our target protein, DNA, carbohydrate, whatever it is. So I choose to address this talk rather to make you understand why basic principles of drug designing we need to know so that we can successfully design the compounds ultimately. However, most often it has been understood that it is a chemistry-driven process. However, if you may allow me to talk on this, I would totally disagree, because what we are making a drug may be chemical, but it will be consumed and should be helpful for biological, which is a human being, maybe even animals. So we must remember that there is a huge biological knowledge which is many times are lacked to see when in computational work or in other cases. And I will show you why we do not get many compounds or many drugs and as fast as possible. Just pardon me, because I will take you back to your maybe grad school or chemistry or maybe chemistry. And I would like you to really look backwards or look at those points. And that's why I put my talk as target-driven drug design. So most often you have chemicals or already known drugs, and then you modify them and find how suitably it works. Many a times you do test on biochemistry, and then you go to the in vivo or on animals or even human for clinical trials. However, the most difficult part is to choose which are the targets, and to choose it not just one cell, one enzyme, but the human being. It's very complicated actually to tell. So my small slide here on the left hand corner or left hand side, which is my the cursor is showing you, shows you actually on two sides, and I have taken it from the online, so you can read it. You can check that online, which shows there are on the right hand side are actual parts of our bodies, and on the left hand side are the molecular, intercellular, extracellular, or different kind of level of understanding, level of where they work. So we are actually now changed our reductionist approach to integrated approach. And that is why the target-driven drug design becomes more important than earlier. So I'll go to my next slide, and which is just depicts all the kind of tools or methods one uses. So and when I call tools and methods, I include experimentals. So there are genomic, where you do genomic functions, and then you predict the genes, and then you go for what are the functions of those proteins, etc. You can develop prediction-based models, like some models with your knowledge of structures. You can as well do little bit more on the reaction based, where you can look into reactions happening at the tissue level. Even you can go for modeling simulations, that is, you can start your proteins or structures or molecules and do the simulations. You can do pharmacophore when you have plenty of compounds already known to inhibit certain targets. You do the generation by pharmacophore or QSR. And most often everyone does docking because they try to see whether the molecule which they design is really good or bad. However, I have seen many of the young generation do the docking first and make sure themselves that maybe that will become a drug. I would like to address you, if the failure is on the top line, then that cannot be a drug. However, best methods on the bottom line you use, you can never make a drug which will be really working at the human level. You may find a lot of inhibitors. So I must make you clear about the inhibition and drug. Inhibition is very important and criterions. And actually, I will talk first of my part of the talk only about the inhibition of enzymes. Very specific chapters. Maybe many of you have already studied it, but still I would request you to go back and study. So I not talk much about the slides or background, but I will try to pursue the topics. So my next is to say that if I talk about target, what should be target? Now let us take protein as model, because most of you use protein. Even the same thing is applicable to DNA as well, in a little different ways, I would say. Now, protein as a target, it will be either enzyme or a receptor, or maybe enzymes and receptors. And it should be in one of these pathways of our metabolic systems. But to become target, its inhibition must lead to a desired effect. Now, let me also put it, though I have said inhibition, because I'm more biased towards bacterial invasion or infectious disease and pathogenic disease. However, let me tell you, it can be activation, or it can be antagonist and agonist taking care of the receptors. So many cases, not inhibition, but activation may or flow in better way. Actually, I will give you an example of that today. Now, but most importantly, it should kill the pathogen. If it is a pathogenic or infectious disease like viral or something, or it might modify some aspect of ours, nor bring back our normal metabolism. And that means functional, if it was functioning abnormally, then it should bring back to the normal case. up to the certain extent, may not be 100%. And the second one is referred to all cardiovascular disease or CNS disease, the neurosystem disease, or even the cancer, certain extent people like to bring it back to the normalcy. However, pathogenic disease, it has to be killed the pathogen. Okay, and you will slowly see why we do not get many of these drugs for antibacterial, antiviral, and why it is so difficult. Rather, it is so difficult. I would like to implore you on that. So there are two already I have distinguished. First, it should have inhibition, of course, and this is a biochemistry led. Second part is not necessarily only biochemistry, and this is where comes the role of target. So let us go through biochemistry-led path, which is why we need enzyme. That's the first question, and I want you to really think second time. Actually, I will give you an example. Most of the reactions in the metabolic pathway, if you study, you will see most of the reactions are nearly spontaneous reactions. They have a negative delta G. Take the case of oxidation of glucose to carbon dioxide and water. Simple reaction which happens inside body all the time. It has a value of minus 686 kilocalorie per mole, which means it's a minus. So what it means? Always it should be, should be converted. So if you give the glucose, keep it, it should convert itself. Whereas it never happens. Why? The reason is here, shown in this curve. I'm sorry if some of you cannot see on the bottoms. I tried my best to push my slides up and that become little congested, but just to bear with you. I was promised that on YouTube, everything should be seen. So I hope who are on YouTube, they can see everything. Here is the right side, there is a box, and you can see that box and you can see very well how this box is run. This picture shows normal reaction should have been, if it was spontaneous, my cursor showed you how it should happen. It should happen like this. No, this is the bookish picture. It doesn't happen like that. It actually have an activation barrier. Hence, you have to reduce this. Otherwise, it will never happen. So the role of enzymes are nothing but to reduce this. Yes.
Excuse me, ma'am. Sorry to interrupt. In YouTube channel, your slides are perfectly okay, ma'am. Nothing is missing.
Fantastic. Thank you very much. Thank
you
very much. Okay, thank you. So what's happening here is the activation barrier has to be reduced. Now it can be reduced by temperature, raise the temperature. So this reaction we do in laboratory by increasing temperature, that's it. But think of how you can raise temperature of human being. Then we could never have broken the glucose in our body. And this is the job of enzyme. So please remember, it looks to all of us very redundant information, but it's not redundant. The reason is in the cell, raising temperature, sometimes the activation barriers are lowered, does lowers. However, that is the physiology of a body. So purpose of having an enzyme is to lowering the activation barrier. That's it. As low as possible, as effective your enzyme will be. That means as fast the rate of kinetics will be as you lower this activation energy. Please remember this. Yes. So this is a summary, what I told already. The living cells always have a limited temperature. They cannot work more than certain temperature. However, there are a lot of pathogenic bacteria, non-pathogenic as well, who work at much higher temperatures. And their reactions are also very different. Also, there are bacteria which are in chilling condition almost, and lower than even chilling conditions. They have a different pathways and different reaction mechanism. However, normal living cells work between the 15 to 45 degrees centigrade. Enzymes are immense important to have all the vital chemical reactions to be done in our normal temperature of body, which is a good range, but still. So how we do the kinetics? Now, here is the equations which you probably have remembered all life, at least in graduate school. So you have a substrate, you have an enzyme, it reaches to a transition state. Please remember, it does not break here. It goes to a transition state and then it becomes product. And these are K1, K2, small K1, K2s. These are flow on one side. That is, if all the reactions could have happened only in one direction, then this will be the rate of reaction. However, it doesn't happen. So sometimes these two reaction rates are different. If they are not different, you can never get enough, but you can get a stable reaction. And stable reactions are most important because that is what all the time we will think. However, please remember this part I have shown you with a stable reaction, that is reversible reactions. But I have not shown you when it degrades to product, no reversible. Some reactions in biochemistry or biological systems are reversible, some are not. And most of them may be reversible as such, but this K minus 2 will be so low that you cannot get that direction. And that is how the flow of substrate or what we call metabolite in terms of in vivo pathway are used and they are made. Basic reaction we must understand about. The reaction limited step is how we go from ES to EP. So if this rate becomes very slow, we don't get enough, we will accumulate more transition states. Also, this is the reaction formula, that is the velocity of flow. Now, velocity is important because velocity of flow decides how the reaction happens. We always assume that there is a steady state, and this assumption may be correct, may not be correct. Later, we'll see these things. Now, it can only be happening if your substrate amount is much higher than enzyme. That is, you have enough substrate, and enzyme is just catching them up and just converting them into product. And these are book picture. You can reread the pictures in the book, very elaborated in the book. You have the breakdown equations are like this. And if you rearrange the equation, we get something called Km. My cursor showed you that Km. Km is most important. And what is the Km, which is, we call it Michaelis-Menten's equation. Is covered almost 100 years back. He did more than 100 years back. He calculated this and says, if we have negligible amount of concentration of enzyme in a test tube and we have enough substrate, then the reactions always go to product. Do you see this left-hand curve? Substrate are all decreasing, products are always increasing. And if you continuously have enough substrate given, always there will be some products coming up. However, if you look at the other two, enzyme substrate transition state is almost saturated. It doesn't change much. And enzyme itself doesn't change much. It just a little bit enzyme released and comes back. And what we call this is called steady state. The reason is ES and E are almost stable. Important to note this because this distinguishes the inhibitors. So if we can have inhibitor against a substrate, then we will have competitive inhibitors. If we have against the ES, we will have something called uncompetitive. If we can have a mixed competitive or non-competitive, where we have partly against substrate and partly against ES. And this knowledge actually is not much reminded of when you make designing of drug and you know, we just assume everything is competitive. Now these two numbers, these two values are different for different proteins and different enzymes, and some cases inhibitors which are uncompetitive may be better off, and some cases competitive may be better off. One of the greatest uncompetitive inhibitor example is anisine. Another interesting uncompetitive inhibitor is penicillin. And third one is Losec, which is omeprazole. So many blockbusters are uncompetitive inhibitors. And many are, of course, competitive inhibitors. Now, we normally, while designing chemicals, we normally take it granted everything is competitive and we just design molecules. Hence, we may not get as expected inhibition constants for every one of them. Now, I would like to go to the next slide because I would like to make you understand about the total amount of enzyme is constant. That's what we always assume in all the biochemical things in vitro, I would say. So we design inhibitors following these equations. These are from the book, so you need not go through each one of them, but we know this is the way to calculate. So Vmax, this is the most famous Michaelis-Menten equation and basic rules for enzyme kinetics and enzyme thermodynamics. More of enzyme thermodynamics than of kinetics. Now, the most important thing here to remember, and you will go to the next slide, you will see that Km can be actually found out by experiments, even Vmax also certain extent by plotting, and we can find the Km is the most important thing because this is the concentration at which the substrate concentration at which the enzyme is functionally doing 50% of the activity. That means at least 50% of activity is done or conversion is done at this point of substrate. Not 90%, not 100%, 50%. So Km has a very important values to consider for designing molecules in future, you will see. So a few more things to remember. One is Km, as I said, and this plot shows you that what is Km and how to find Km. We can have a turnover number. This is also important because this is how faster the reaction can happen is measured by the turnover number, and it is dependent on Kcat, how the catalysis happens. Many a time, this efficiency, catalytic efficiency, which is defined by Kcat by Km, and the turnover numbers are important for receptors. Hence, I would not address that so much, but please do remember this. Well, this is all about the basics of rules. What about the inhibition? Actually, there are, as I said, there are plenty of different kind of inhibitions, and most of you have seen in the books and journals and papers the term called IC50. Normally, IC50 is not written like this. IC and 50 is a subscript. But I will come to this point next. Actually, one should look into not IC50, but Ki, which is actually Ki is the inhibition constant. So I'll come to the relationship between both, and it's important to note this. This is a Chen-Parsov's equation, which is published in all back in 1973 and modified later on for different purposes. These two, that is IC50, is not necessarily directly measures the affinity of binding. Here I would like to bring to you all to remember, all you calculate the docking, binding values, delta, delta G, etc., has really nothing to do with IC50. Sadly telling you that. Many papers I've seen, I've reviewed, people use IC50 to correlate with everything else in the world with free energy calculations. Basically, you should not use IC50, you should use Ki. And Ki is the most uniform inhibition constant which specifies the property of actual inhibitor in the substrate. Please remember this cautionary values to you. Now comes what is Ki and how it is related to IC50. Generally in the, everywhere and even Wikipedia, I thought best is to follow Wikipedia because all of young people are very used to reading them. And I figure out they have correctly estimated values. And this is the Ki. As you see very clear, would you see this very clearly? Ki is related to IC50, but also a function of substrate divided by Km. That means if IC50 is the strength of the inhibitor, it may be functional strength, but it is not actual inhibitor. And IC50, that IC50 will depend on, I'll come to that also, on many factors. Now this is for enzyme inhibition, whereas Ki and relation of IC50, if you look at the something like agonists, receptors, this is the equation what you should use. And please do note both the cases there is A and S, and these two are the notoriously changes everything in IC50. Hence, what I want to make you sure about one more thing is IC50 value may vary. It's not really a constant. And it will vary with how you do the experiment, what kind of substrate you choose, what is the concentration of enzyme, etc., etc., etc. So you don't compare one IC50 experiment with another IC50 experiment. Please be cautious on this. So, and the reason is very much scientific. Rather convert them into Ki, but Ki also has a bit of reservation. This inhibitions, Ki's is really good absolute value. Everything is, and all are in molar concentration, molar per liter, etc. So please do remember these units are all decided in advance. But if the definition is perfectly okay, that is competing ligand, if there is no receptor at all, then this is what 50% would occupy. 50% of the receptor will be occupied by the Ki at that concentration by the inhibitors. Absolutely right. However, it's not necessary always it will work, because we have seen this effect. We have seen many cases. If these relations, which I showed, may not be tallying, may not be correct. And in those cases, you may use different reactions, different relations to convert IC50 to Ki. In those cases where the inhibitors rather is tested at very high or very low substrate concentration. Actually, when the inhibitor is too good, that means nanomolar, then this relation doesn't work. And if the inhibitors are almost molecular, mole level, 550 molar, this relation doesn't. So please remember this also. And this doesn't, the reason behind is very much to do with what I said in the initial stage. We take all reaction, enzyme is very low and substrate is very high. And substrate can never be in scarcity in the cell. And all this not valid when you have that kind of reactions. So please do remember arbitrarily or a lot of things to remind of yourself when you use IC50 and Ki as if they are similar things. Here comes the crisis point, I will say. Yes, here I'm trying to show you the case study, which we did simulate it, but nonetheless, if experimentally data available and plenty of papers available in early 80s and reviews have been done where you can see these discrepancies. Now, let me tell you what is that. First, I want to introduce you that there could be 3 types of inhibition, and which I showed here. One is competitive, one is uncompetitive, and other is mixed competitive, means both and which I have already spoken to you. If it is inhibiting the substrate, then the inhibitor substrate, it goes to the cavity where the substrate binds, then this is called competitive. In many reactions, it doesn't do that. Many inhibitor for the same enzyme may go to this complex, bind to ES. That is called transition state complex. And then the reaction would be uncompetitive. The formula has to be changed a bit. Formula has to be changed. These are the velocity through passing through. Okay. And it can be mixed also, where partly inhibitor goes to occupy and partly it does this. It can happen. Some inhibitors for the same enzyme can behave this way. Am I clear to all of you? But while designing molecule, you only remember this and just forget anything else. However, when you design molecules, you may see some acting assays are perfect. You get the same competitive inhibitor. Then you modify the compound and you get totally different results. Why? You are surprised why it's happening. It should have been, I increase the hydrophobicity, so it should have been better binder. It becomes worse. The reason may lie that your molecule changed its characteristics from competitive to uncompetitive. And you must do a biochemistry with that separately instead of only doing the results and taking the assay to find what is happening. This is another cautionary note to all of you. And this happens because same target can handle the inhibitors in very different way. Now, this is a simulation we did for a long time back by a trainee, where we took, let us say, we have a reaction, many reactions maybe, and there are 3 reactions at least, KM-S1, S2, S3. Let us say that all the reactions are happening in the same Km value, and their their products rate is also the same, etc., etc. Okay, substrate rate is same, product is this, and the inhibition, if I want to inhibit them, is Ki, which is millimolar, and the substrate concentration, etc. All of the substrate concentration are almost 0.1 millimolar, and Vf and Vr, which are the flow through, are these values. If we assume all these three reactions happening, and then we plot all the 3 reactions with Ki being 3 of them. That is, I can go back, competitive, uncompetitive, and mixed competitive. How the curves will behave. I will only show you competitive and uncompetitive cell when only 1 reaction happens, not 3. That is in vitro you measure. And when all the 3 reaction happens, which I call in vivo, all the 3 reactions are happening and each one of them are exchanging their substrate. They may have an opportunity to exchange their substrate. Okay. And this is what the in vivo versus in vitro I noted here. I'll show you simple simulation plot. Here are the most interesting plots are uncompetitive. So if the inhibitor is uncompetitive in vivo and in vitro, both cases, we use the equation I showed you earlier. In vitro, it should plot like dark blue line here. So your IC50, which is you can get inhibitor constant. So IC50 will be around maybe 0.015 uncompetitive and in uncompetitive in vitro, and green curve it will be little bit 10 times, not 10, maybe 2 times more, 0.25 in here. Fine, it will be 50% around here. However, the 50% is here. However, look at the competitive inhibitor. Competitive inhibitor is in vitro, 50% is also known, and this is in vitro, that is the magenta line. And magenta line in vitro comes here. It is almost 0.025 or 0.2, which is quite good inhibitor. Okay, so you go from biochemistry, jump into in vivo, that is in the mouse or in the cellular milieu. So you put the compound into the cell and expect that you should get 50% at the concentration of 0.02 or 0.25. What you see actually is 50% concentration. Actual in vivo, it happens to be not even reached, but you can have it 0.2. Here is the anomaly. We did publish also these papers. It is the great anomaly is here, and the great anomaly only tells you that when you are working in vitro, you may get compounds acting fantastically good and very low IC50. However, you go to the competitive, that is in vivo, it fails. And this is major failure for when you have competitive inhibitors, whereas it is less failure for uncompetitive. It is only 2 times as I showed you. Please remember this story also. So when you design compounds, please do remember, and all these happens because target is enzyme. Target has decided itself what to do in advance. That's what I call that honor the targets, and we don't care, we just take these values. But actually you must be careful designing competitive inhibitor, and that is the reason. Huge failure in drug designing of against the main bacterial, or even if you think of human disease like metabolic syndromes. It's not the chemist failure, it's not the biologist failure, it is actually the difference between the biochemistry and biology. Now I'll go to the challenge. So If we want to design a drug or something, new drug, antibiotics, etc., where should we go? So our challenges in compound design, is it the challenge is lying with the compound design? No, I don't believe that is in compound design. I think enough designing tools and designing methods and synthetic chemistry, very strong, contributed last 50 years. Many major breakthrough has happened and lots of new reactions has come up. So compound making is not the challenge actually. Challenge is who will be the targets. And that's why my rest of the talk will actually deviate from that. My full dedication will be why appropriate target is important. So, yes, am I talking something else? No, there is publication and you can read this paper as well. What are the targets? Targets I have defined earlier. Actually, what they did is, these guys did it, that they took all the known compounds already been known to be drugs and associated drugs or something. They figure out only 482 proteins or associated targets. Some of them are well-known proteins, but associated targets have been identified. So available chemicals means drugs have only 482 targets. Human genome says you can have 8,000 targets. This 482 includes bacteria or human, both, and bacteria, virus, etc. plus human. Now, potential hits, what you call, like around drug-like substance are nearly 5,000. Antibodies are 2,400, 800 protein pharmaceuticals. This is the year of 2006. If these numbers have increased almost double. Now the question is, so how can we predict? We predicted, they predicted 399 molecular targets possibly, which belongs to 130 protein families. If you consider human base, human data, then these are the numbers, and rest of 100 odd are for antibacterial, antiviral, etc., and anti-malarial, etc., all those pathogens. However, just only 399 molecular targets, unique proteins I call it, and they belong to only 130 protein family. So practically we have 3,000 targets left out, which belongs to 218 class. So there is a great scope of finding these new targets. And why new targets? The mechanism of action can be started. My last slide will show you how best you can find that. However, the rest of them are more or less of talking about how we grown through last decade or 2 decades or so. So plenty of targets research can be done. Now comes, okay. So it's not only just should be having effective, but we have to identify those enzymes or those proteins, if we're looking at only for protein molecules, which are essential metabolic pathway. That is most important. What is essential metabolic pathway? That should be available in the if it is a pathogen, it should be available in pathogen and should not be available in human or something or other like the host. If it is important, that is, if it is a human, it should be available in the pathway where it is to be impaired. That is where I should interfere with it. That is that particular pathway should be studied to find which protein is essential. That means there will be some proteins which can work as a slow protein, and there could be many proteins which can work as a very fast protein. We have to choose which ones would be good and effective for us. And this is how one can do, and this can be done by the methods I will talk later on. And this is important. The reason is every selective, or by doing the selection of proteins which is available not in bacterial and pathogens but available in human, or just available in human pathway because we are looking for a diabetes pathway, or we are looking for plenty of enzymes, you will see. Now, just because it is crystallized and just because some data available, will it be called as essential enzyme? You don't know. But we do manage to make a lot of theoretical work, and then failure comes because they are never gone to the actual pipeline of drug. The reason is because we did not choose the target. Now, how to choose? I'll come to some of these. So please remember, essential is important. If it's not essential, will not be working. Second, I will not give this example at all today, but I will mention to you, the inhibition should not be that you put molecular level, that means mole level inhibitor to inhibit the enzyme. And that's why I already spoken to a little bit. If the enzyme very fast, then what happens? You go on pouring, it will take inhibitors and just stop the reactions, and everything will be converted. Because you require 100% in his enzymes to be inhibited. However, even 1% of that enzyme can produce the pathway and continue the pathway. So this is one other question, is you must not put too much of substrate, or sorry, the inhibitors, so that it becomes toxic. So it should not be in the millimolar level inhibition. Then you cannot have the molecule gone for the next level. However, I will not much talk on this, but I will try to elaborate how in silico and in experiment, both you can identify that. Now, yes, this is the IC50. This is endogenous method. That means this is a disease. And this disease is only IC50. So most of the endogenous disease or metabolic disease, you can trust much on IC50 and convert into Ki for your calculation. However, please remember, you are bringing back to a normalcy. You are not zeroing the pathway. You actually allow it to be normal pathway. Hence, designing compound from biochemistry directly to go to pharmacology is easier in this case. However, in case of receptor, you face the same problem what you face here, which I am asking you to look at. When you come to the pathogenic or infectious disease, even cancer falls under this category. You require to annihilate the whole inhibition. That means you need 99. You don't count 50, I say 50. Hence, compounds you might have produced, which is biochemically correct, and maybe the same will be effective. However, 99, you cannot get it because that will take the slope of the curve. If you remember my earlier plot, the orange curve, it becomes very slow after 50% inhibition. No steep going up. Hence, you have to put earlier, as I told you, maybe 0.25 was your good inhibitor for IC50. You require almost 5 micromolar compound to be successfully making them. So which is 100 folds, more than 100, 200 folds increase in molarity, and that causes toxicity. Hence, many compounds have been identified and as biochemically and conveniently and very good, well assessed, fails because of this reason. We need to go for 99 for pathogenic systems or even cancer. However, for other purposes, can be IC50. Now, how to do this? We can do 2 ways. One is flux through essential metabolic pathway can be decreased. So you decrease it so low that ultimately nothing comes out and it becomes stop and bacteria cannot grow or virus cannot survive, etc., etc. Or you bring it back to control. That is the amount you control so that too much flow is reduced. Unfortunately, for time being, I cannot discuss much on this. We had some of the targets identified by this method, but I'll just hint. Another thing you can do, and this I will not also go, but we did suggested a method by which you can actually do this. That is, I'll come to this, how you increase the toxic level, because you are stopping the enzyme inhibition. So your substrate is becoming higher and higher concentration because the previous reaction is happening in vivo. In test tube, it does not, but in vivo, it happens. Hence, your substrate can go higher and it can become toxic if it is accumulative. But if it is already been used up as it happens in a metabolic pathway, if it is used for other reactions, then you cannot increase that. So you have to be very selective to find this. Okay, go to the next. So what is the problem with pathway analysis? Why can't we just take pathways and do it? If you want to do, there are many methods. I'm coming to them, but it is quite a tough work because every pathway are different. So if you try to address them by graphical ways, it's a bit difficult because everywhere you have to use your own different kind of graphs. If you want to use for all of them, it's really difficult to calculate every cases by the metabolic control analysis. However, there are other methods by which you can actually look at the whole system also. So these two pictures gives you another, very many are there. Now, why the difference in vitro cannot be linearly translated in vivo? And that is because as you take a pathway, let us say one of those old model pathway, as I said. Now let us say if we do not inhibit reactions, then what happens? That everything flows and it produces S4. Okay, the S4 is produced. However, if we interrupt this enzyme, we inhibit this enzyme E2, what happens? Two things happens. S3 goes reduce. S4 effectively goes. The enzyme is available, but no substrate. So it also goes lower and lower. Eventually, S2 accumulates because it cannot be utilized. In case it cannot have any other reaction. Many cases it might be utilized in the cell. However, it can happen that it becomes toxic because of accumulation of this. Or cell cannot function, but because S4 was very important to the cell, and this particular molecule may be an amino acid, may be converted amino acid, which should be in the protein, and it cannot make the protein. So it can be very important for that. However, in a vivo, just this reaction doesn't stop because you stop this reaction. There are other pathways who are supplemented by this particular S4 metabolite. It can come from other pathways. Just like this cannot be toxic because it can go to another pathway. So all these S2 and S4 modulation happens because of enzyme E2 to be inhibited. However, can we estimate these a priori and find, if we know the enzymes characterizations and we know the substrates reactions, we know all this, can we a priori do it? Yes, we can do it, and has been proven to be done by long back by a group of biochemists, and they develop tools also, which I will come next. So this is what I just now told you, and it can be done, and it's called metabolic control analysis. There are other methods, but I would not discuss those methods also. But however, I will just say them. So what we can do is we can calculate, these are the functions and these are the coefficients. The coefficients are like this. So if we calculate each, how quick the coefficient goes, and this coefficient defined here. So log of flux is flow of the velocity of the reactions, flow of the metabolite, and with respect to every enzyme's concentrations, we can calculate, we can actually find decreased flux or increased substrate, etc., by using this formula. And you can actually measure this, whether my theoretical prediction is good or bad. You can do it indirectly by knockdown experiments, which are molecular biological experiments, and I will tell you some recent reviews as well as I will tell you application of it, wonderful application of it, very recent has been done. So which is experiment and theory together goes in hand, and particularly this is a field. As I told you, 3 different ways you can handle these problems. One is metabolic networks, which is metabolic networks. You actually do the calculations, which I'll come in details. Another I will not address, one is flux balance analysis, which is not to calculate each and every reaction, but just the fluxes and solve them, assuming a few more guidelines. And some of the works we did with tuberculosis with the flux balance analysis, though I will not discuss much here. And third model is, in this case, you can do the whole system. Last one, what you can do something like this, like pathways, isolated, but You can do in human very easily. The reason is it's like using graph. It's like logical modeling where you do not go, real values of flow. Rather, you allow it to yes or no flow. So that's why we call it logical modeling. And it's been very used in signaling networks and pathways quite well. However, I'll give you some examples of only this and how to do it. Earliest possible papers have been cited here, and actually it is a proceeding, and even the papers have been published. You can search by these two guys' name, by Ethel and Maria, and both Ethel and Maria was the people with whom I discussed and learned my topics, what I did future. They worked on trypanosoma glycolysis. Actually, Glaxo was interested to find a drug on trypanosoma glycolysis. And they did find many compounds, and all they did is biochemistry of each one of these proteins. And if you look at the glycolysis pathway, these are the set of proteins or enzymes are available. Each one of them you can see. There are plenty of enzymes, like you can see the phosphoglycerate kinase, triphosphate isomerase, hexophosphate isomerase, many of them. And all these structures were known. Glaxo did for almost 10 years struggle to find compounds, which is fantastically IC50. Good Ki in test tube, but if they put it in the actual trypanosoma cell, it does not inhibit the growth. And here they learned the lesson, and I learned the lesson from them. And that's the point. Is there are few important things to look at when you design the pathway. Entry of glucose is most important. Now, if we stop the glucose entry, probably that is the best. But how you do it? Trypanosoma always will survive in some food, okay? So you have to deprive it food? No, it will survive with the foods. However, there are cycles where there are phosphorylation happens. There are breakdown of phosphate, which is here. There are conversion of pyruvate, which is at the end of it, and release of pyruvate. So what these guys were looking at to generate all the equations, they included even this part. That is, which is in the mitochondria, because this inputs certain amount of glycerol 3p and dehydroglycerol. So they did simulate the whole and identified wonderful thing. That is, they said, I'll show you what they did actually, but let me give you the final point. What they figure out, you cannot stop all these reactions. However you inhibit, you cannot reach the inhibition to stop pyruvate to generate other than glucose transporters. So inhibitor should be designed against glucose transporter. Now, if you know a little bit pharmacology, you may think of, actually, there are compounds available for diabetes, anti-diabetes, which stops glucose transporters. They modulate glucose transporters. And practically they found that drugs are perfectly working against trypanosoma glycolysis. So here it is a point. Is that my time over or something? I hope I have few more slides to go. Oh, nearly. Yes, we should be looking at these these papers more thorough. Now, what they did is they did a few reactions like this, and aldose is very different inhibitor, so they had to do uni-bi substrate reactions. So they did that, and they did include some conservation constraints, and they found what I already told you. The results, which I have not shown here, but you can look at the papers of Ethel's. By search by Ethel and Maria, you will get plenty of papers around 2004 or 5. If you want, I can supply you the papers as well. Okay, so how we do the modeling? This is the computation part, which most of you are probably eagerly waiting. And these are the softwares available. And as I told you, recent software publications is also there, which is 2010 versions available here. I actually trained some of the students on the software in May, middle of June. Some of the students who are taking my courses on system biology, particularly on MCA calculations. So I offered them certain things, and they did work out They are solutions of competition tools. How you get the parameters or the reactions. This is how you get the reaction. These are those fellows, and some more can be included. How you do, what is the pathway? Selection of pathway, and like this. Then generate the equations, which is perfectly whichever you want, and reactions can be generated by this or Brenda. Then some data which you can get from KEGG, you can get from Brenda, and a lot of published literature you might need. Then solve the equations. There are techniques to solve the equation, which is incorporated in Zernak and Kokasi. You can use Zernak for solving many this type of equation, mostly differential equations, single differential equations, and analyze the input. And that has been done by R or by Python, whatever script you like. It sounds very easy on those, but do you have, should I do the target searching for every one of them? I would suggest you can choose the pathway and then do for all the enzymes and try to see who is the pathway blocker or who is the most essential in that pathway. And then take that protein to design drugs. That is, you can find whatever method you like. If already known compounds, do QSR, pharmacophore, etc., do docking. You can do anything, but identification stops here. Is this good, bad, ugly? Can you do it? Yes. So we did actually PhD thesis of a student of mine and 2 PhD. Another I didn't show here. He did with signaling network inhibitions. That's for human, and particularly Vivek's thesis I have followed mostly. And he did that. This is the topics which I am going to detail little more, 15 minutes, maybe more, and show you how we were fooled. Instead of inhibitor, we actually try to do something else. Realize that we have to try something else. And this is because purely we did. This was done by Vivek and found out this. And it will be discussed more because this is how to stop dormancy of TB and address the TCS cycle and shunt pathway. We did also work with this concentration increment. That is, I told you that substrate will increase. And if the substrate increment cannot be utilized by the cell, it becomes toxic. This is another pathway we found, and anti-malarial target we identified. We've got it published in FEPS letter or somewhere. Maybe papers are coming. My papers list are also here. Yes, FEPS letter. And last one is in thesis of essentially full TB genome, which is work by Vivek, and later on followed by some other students in JNU. So we are trying to publish this paper. This is already been published, a list of very essential genes, and even AstraZeneca started using that profile to make more anti-TB compounds way back in 2003 to 2007. Well, last 2 papers are the most recent papers which has utilized this metabolic pathway. One is Kisling's paper, which is very interesting, and they have used wonderful method. Actually, what they have used is they use genome. That is, they have used CRISPR-Cas control and experimentally found what they predicted theoretically can have good blocking and good cell survival in yeast. Another people, and this is also mevalent pathway, how it can produce much more substrate impact. And the last paper, that is even more interesting, which is published just recently, 2020, if I'm not wrong, yes. And they have found the binding of these phosphorylation, this is a network, but used control analysis, that this is a neurochemistry, neural system control can be handled by using the same thing. So as you see, this field is growing. People are trying to identify targets. I'll come to now a few slides of dormancy to stop. So don't worry about all reading. You can leisurely read all these because you will have the on YouTube. What I try to show you here is only very interesting points. Point is this is how the glucose is broken and goes to the oxaloacetate. What normally cell does is it follows the main pathway, which is written in blue, and follows the whole. However, the E. coli also follows the same, and many other bacteria also follows the same. However, if the cell doesn't always get good food, that is, it's not, you do not get good food. I know what happens, like during this hostile situation of lockdown, you don't get your favorite food of Chinese buffets. What you do, how you survive? You eat rice and sambar, maybe rice, rasam, and curd rice. Maybe some North Indians, they use chapati and only curry. And some Bengalis, maybe, or Orissa, they might be eating only rice and a vegetable. Why? Because your favorite food is not available, but we learn how to accumulate different ways and different things. Imagine a poor small bacteria. If you deprive it with its normal food called glucose, what it will survive? It cannot crawl out of something and get its food. You are not allowing it to have food. Hence, what it does is it uses its excreta, and that is acetate. Many reaction happens which creates acetate, and acetate normally leaks out because it's not good for the cell. However, because in the diverse condition, bad condition, surviving condition, it starts creating acetate. It uses the acetate, and if it uses acetate, it's a big deviation. Just to remember your biochemistry or chemistry module, glucose has 6 carbons and acetate has only 4. Now we lose carbons here, 1, 2. So if it's a 6 carbon, then we can lose 2 and go back to 4, which is oxaloacetate. However, if we lose these pathway 2 carbons in acetate, then we don't get that. We become what? CH2CO2. We don't get anywhere oxaloacetate. So bacteria behave in a different way. They got a shortcut and called shunt pathway, where from here a substrate or metabolite isocitrate can be utilized other way around by few proteins. These are the genes shown, and these proteins are called ICL. This protein is called MDH, malate synthase, and these proteins will convert glycosylate, malate, and oxaloacetate, so that it does not go to this lavish pathway to lose 2 carbon dioxide and cannot have the reaction after this. This is inbuilt because they do survive inside our lungs where they do not get glucose. Tuberculosis do survive. Even some of the E. coli pathogen do survive in absence, not in lungs, but in stomach, in the guts basically. Many other things have been done. So we actually produced this model and took a patent that many of these intracellular bacteria can have stoppage via this pathway. So this is a way to stop them or deprive them to go to this. So the condition was that we should find a easy enzyme inhibitor of this ICL. I'll come to the story in a very nutshell. It was found out, and this inhibition was done by the groups, and we were in close contact with these groups who were making. They did inhibitor find ICL block and tuberculosis macrophage they found, but this molecule needs to actually stop the macrophage. They need almost 10 micromolar compound. And as you see, if it's 10 micromolar compound, as such, this 3NP is a little toxic at very low level itself, and it will be worst. Now the question came, why don't you compound, you design many? I would not give that hints to any one of you. Because I want you to read this paper and see that what the molecule 3NP is, the chemical, hardly have opportunity to modify anything. This group were almost disappointed, said, why the cell does not get reduced? Why can't we design a smaller things? So they gave up. ICL structure was found out after that. After that, structures were found out, and they tried to design inhibitors in a large active site. None of the work happened to be successful. They found inhibitors, but it did not kill the pathogenic, what you call dormant bacteria, which are hiding in our lungs, and uses acetate as substrate of their life than the glucose. So this is the story, as I told you. Why we need? Because lengthy chemotherapy is because of dormant or persistent tuberculosis. No drugs available at present also, except it was found that newest that bradiquilone might be effective, but the pathway nobody knows. But at least recently that has come out to be somewhere killing the subject. Then resistance to almost all drugs, slow grower bacteria is this type of bacteria, and fatty acid as carbon source. So all these acetate are having this fatty acid carbon source. So this shows you that glycoside bypass is essential. So we should able to. However, the failure is very blatant. So what we do? Hence, we took this up and did the simulation. I wouldn't go detail of this. You don't need to read. You will read detail when you have time. There are many pathways. E. coli has some other, and bacteria has some other. So our idea was we should inhibit ICL. And if we inhibit ICL, what normally will happen? Normal cases, when bacteria goes as dormancy, there is an, at least it was found out by E. coli long back, none other than Ghoshland, that isocitrate, there is a isocitrate kinase, isocitrate dehydrogenase kinase, and that kinase actually deactivate that ICD, which converts ICD into alpha ketoglutarate. So that enzyme number 3 can be inhibited, not to allow the flow. But then this particular enzyme has many structures, hundreds of structures. Actually, ICD has a many subclasses. Plenty classes are similar to human. Bacterial classes are little different, but active sites are almost similar. Hence, ICD inhibitors would work for human, and human already been identified. Many human being who have a depriving of ICD, lower expression of ICD, gets very different kind of disease. Hence, just to inhibit ICD was not a sure story here. So here is the default of choosing ICL and choosing ICD. So where we are, we decided to do the whole simulation and find why ICL is failing. Instead, we decided, why don't we look into the tuberculosis module and see which are the kinases? And we did do kinase identification, etc., etc. Whole story is very different. We have got some patents on it, and then we tried to find, can we diversion made pathway? That is, instead, when it is normal case, the pathway will follow this, but well, the most important is that eukaryote doesn't have this, so we can ignore eukaryotes. Doesn't have shunt pathway. And another case is when it is very much less, that is only a few palmic acid and just acetate, then we lose the carbon. So what we do is, instead of following this pathway shortcut, so we will have inhibitor design here as well. Stop these not to make ICD inactivate. So we identified the kinase which makes inactivation of isocitrate dehydrogenase of MTB, and can we stop that kinase? That would be the ideal, because then what happens? The story is very simple. Not only will make ICL, but we allow everything to flow this pathway, which is a futile cycle because 2 carbons falls off and nothing goes after this. So dormant bacteria can be really stopped because they cannot produce survival. They cannot survive by using even the minimum. Of course, once you bring them back to normal glucose, they will follow, they will go. So this compound, which will stop the interaction between these two, is this possible? Yes, we did circulated all these, and I'll show you the target. And so that aim already been told you many times. So we did so many simulations, and what are the results? And that's what I would like to share with you. So basic premise was by diverting the substrate different pathway. That was our goal. Then we go to the steady state solution and many of those parameters has to be done. This is for those students who are interested or participate who are interested to calculate. Please do remember, you must do this kind of simulation and experimental comparison, and be careful to compare proper functions of every of these are the metabolites. So we did that, and then we said what we do is switching on the glycosylate flux instead of shunt flux. What we simulated, what we got, the results I will share with you. What we observed was both ICDs has to be inactivated. Otherwise, the flow to ICL will not be sufficient, which is tough because we cannot do selective ICD inhibitors for human and bacteria. Bacteria has, particularly MTP has 2 ICDs and 2 group of ICD. One ICD is non-eukaryote, the second ICD is very much eukaryote. These studies are done by some other PhD students of mine. So inactivation by this kinase is already shown. So we have earlier papers, and we did experimental data also with this. Ourselves shows that inactivation does work. So we propose the possibility by this inactivation. This is the table. Here I show you the flow through ICD1 and ICD2. So there are 2 types of ICDs, and both the ICDs flow. What you notice here is that the flux through the ICL is given by the glycosylate bypass. Bypass is the shunt bypass, and this is the normal. So what we see, the fluxes are very high in this if we have even any amount of flow of ICD is lowest. But it is not so in the case of ICD2. So ICD2 inhibition in this part, in the first part, very low flow happens when we don't inhibit ICD. So ICD flow influences ICL. That means if we do not inhibit ICD2 enough, it will flow through. This pathway still will work. What happened? I told you earlier to remember competitive and uncompetitive inhibition. Here we show you ICL competitive and ICL uncompetitive, because I gave you an example how the failure, they could not really find how to solve that problem. So what is our next slide will elaborate a picture. Efficiency of competitive inhibitions of ICLs increased with the increase of ICD. So more we flow this redundant or futile pathway, we force them to go more on this pathway, then the product of isocitrate to go, least it will be, so more and more it will be inhibited for competitive inhibitor. Uncompetitive inhibitor is not so. Here is the choice. So we require a competitive inhibitor for ICL and inhibitor for kinase so that it does not inactivate the ICD1 and 2. Here is the data shows, here is the data, you can find out this, you can read it by yourself slowly and see that. So you can see these effects and plots, how to plot also is shown here. For example, uncompetitive inhibition, already I told you, it doesn't influence much. What happens to mixed inhibition? If you remember, 3NP is a mixed inhibitor. Actually, mixed inhibitor was the most tiring problems. What we saw, it's the values of IKI was very poor, and not only that, what you see is almost we reproduced that. There are 2 ICLs. So what we reproduced is that almost this much millimolar has to be given to calculate the Ki for ICL. That's what our calculation shows. So obviously it is quite a lot of compound to put in. So mixed inhibitor also blocks growth of this phase, but high concentration, and which is more like what we saw as you see these values. So this justified or sustained our fluxes, and flux reduce is hardly any. So you see, if we use the flux, we can reduce the flux only 70%. So lots of still ICL pathway, lots of molecules will become oxaloacetate and cell will survive. And that's why cellular milieu, they were not surviving, whereas laboratory, they were surviving, and macrophage, they were having lots of problems. So this was the final strategy, as I already said, and there are advantages. So we can have a competitive inhibitor of ICL. This is what is suggested, and we can have ICD kinase active with the competitive inhibitors for isocitrate. Both of them. So kinase should be inhibited, and as well as the competitive in ICL. This has been designed. However, we are working with this part of it. This is a protein-protein interaction, but we are trying hard to get that broken. If possibly, we can make that. Then comes advantage. The competitive inhibitor, that is one thing, and sufficiently to stop the flux through the axonate. So problem started with to find some target, and we thought just quickly we'll get it, but unfortunately it didn't. This is a summarization. Hence, you can be happy with, at least I show you summary. Next slides, 4 slides are there, and I would show you standard protocol, not so much cumbersome mathematically and doing identifying things like which targets are good, which enzymes will do the job and then go after it. But there are simple rules, and there is something called, this is my take-home summary for all of you, that we are designing this. This is what we have worked out, and almost we have found the compounds. We are at the level of finding compounds, and we have found compounds, but we have to test assays to have this interaction. At present, we are doing that. So how to build a target? You know, after talking so much, I would like to give you some minimum criteria how to do this. You know, it cannot be just something funny. You know? So these criteria are really validated by very many people and came out with. So how you do this? Universality is many species, but not the host. Essentiality is many species, but not in human. Of course, it should not be here also not human. Vulnerability is which out of those essential enzymes, which ones we should choose. Then feasibility, that is whether we can design molecules or not, crystallization, etc. Actually, once you identify, I think this becomes much easier nowadays. And then you can find the inhibitors or design inhibitors, and then you can do with the chemical properties happily. So these few characters, 1, 2, 3, 4, 5, has been actually incorporated in the soft, some online, which is dedicated for unmet disease. And we worked with this group for a long time, and we offered some courses on how to do this hands-on, and I did that, and so I show you. This is in 2009, and they have developed this. Basically, this was done in 2008, and this whole unmet disease, a wide range of data. The assessment is in silico, and it uses, if you do not have that particular species, you lack that protein, you don't know whether the protein's data available or genomic and genetic and biochemical data available or not. It uses orthology based and finds the functional evidence. So this works, website you can go, and it looks like one of these. Recent version is like this, though last 5 years has changed everything. It has become more easier. However, now it has become a little more difficult. So please do read this manual, which is resources. Under resources, there are manual and tutorial. Please read the tutorial and manual, and then only work on it. You would be able to find target, or you can find drug. So if you can find targets, you can go to the chemical, and it will search the chemicals and also gives you the chemicals. So this has been quite an exciting progress, and the version here has been. Now I come using this kind of techniques. I will come to the point of, because I exhausted my time, already exhausted almost maybe 18, 17 minutes, but another 3-4 minutes. Using these techniques or these procedures, there are drugs already against COVID. COVID-19 is the biggest challenge to biologists and chemists and everybody, all scientists, all features. Even modeling becomes very difficult. Hence, we did some study on it, and then I did take published papers so that you are feeling very comfortable because lot of published papers, non-published bioarchive are withdrawn later. Hence, I will show you systematically two different approaches, and you can do any of them. But one is to find the targets and following those criteria. So if you look at the viral genome, it's not a very big genome, 29 proteins. Hence, you can have what we figure out 8 proteins actually serve, makes this feasibility, druggability, everything satisfied. And not only that, what we notice, not only like us, we design or we do something, what we notice, there are people who have already published paper, which is here, March published paper. They actually identified the proteins and they did, some of them crystallized. But what they did is, they did a what I call homologous study. That means those proteins are available in other viral systems who belongs to the same group of virus, corona group of virus. And this coronavirus earlier is HIV, there are Ebola, there are SARS, and they designed compounds, or rather they did not design, they actually repurposed compounds and they categorized the compounds. And this article is really interesting to see. These are 3 of them already known viral protease, and they reconfirm that they are working in the case of COVID. These 2 are, one is protein expression, such a small, very good molecules, protein expression, and this is for a viral surface protein inhibitor, which is actually a compound is now synthesized, and many model out of this has been. Many of the medicinal chemistry is worked out with this and this now. And RNA dependent, which is most interesting. There are these compounds which are RNA dependent, RNA polymerase, which is very much homologous here. And this is very unique. However, some of these compounds may work here and people are trying experimental work to prove these works or not. 4th category they figure out is which is viral infection. And this is where the HCQ, this is where HCQ plays a role, because infection, it stops, and it stops in a totally different way. It actually interacts with the host and the viral interaction. It interrupts that. However, every mechanism of action is not known. Most of the mechanism of action known for other virus, not for this. However, these are all gone to clinical trial to identify. This is, I would call, typical way of finding as quickly as possible chemicals to interrupt the disease. But more interesting paper came out in July, and the work was submitted quite long back, and I was intact continuing with them. And that I found to be more attractive. So my last slide contains that. So how to find target is, another way is host-guest relationship. What they did is this is the genome and 29, this is from the paper. Genome 29 for structure and these many proteins are available. So viral proteins, 26 of them interacts with 332 human protein. They did it in experiment. They did a massive experiment in different labs, identified every of them uniquely, 26 of them. And what they did is cluster those proteins interacting with which one of them. So they did the graph network also. Beautiful networks are shown in that. And most interesting is, as you see, DNA replication, these are human, has this interaction. Epigenetic and gene expression regulator, which is in the human, they interact with these ones. So it's very interesting how these proteins are interacting. And ORF, these are accessory factors. They call accessory factors. These are also small proteins. And all of them seems to be working out with some other places, like they figure out sigma 1 and sigma 2 of human has got very much strong influence with one of these. So now the target is not just one enzyme of viral, but interaction interruption. It's something sounds like earlier I showed you. Invade protein-protein interaction. And one of the protein, they have actually known crystals. They identified these are the potential binding site. Rest of them are not. So the protein of the virus can bind here, these places. So if they find molecule to interrupt these places, then the protein, these fellows cannot bind. So what they did is model some of the proteins. Some proteins they downloaded from the crystal structures, and extensive operation they did with known protein, known inhibitors, and already gone even to preclinical or drugs available already. And 69 of them found to be very good. They worked with Brian Shaw's group as well to find by chemoinformatics tools. 69 of them were identified that they can interrupt these reactions. Out of the 69, 47 compounds found to be, they include in all these, found that they modulate 62 protein. These are human protein. This is the best thing. That means they are binding somewhere here, one of those. So these compounds will not bind to virus, but it will bind to host protein and don't allow the virus to interact. Supposed to be the best way to find the antiviral drug. And I'm not sure why people didn't try this earlier. Apparently they gave references, some of them Kogan's group did with HIV, something like this, and did find something else. So this is my major part of the talk. And without these fellows and these fundings, I wouldn't have been working for so long to survive. And thanks to all of you for doing such a great job, and to listen if you have. And anyway, you must see this, that you can have many more question answers and go through it later leisurely and see that you are ready to do that. Thank you very much, the host and Imanchi. Because of them, I could manage to go on live.
It's okay, ma'am. Fine. We have received few. First of all, thank you, ma'am. It was a very overwhelming, informative, and conceptual session indeed. So we will like to take few questions quickly, ma'am. So may I start question answer session?
Yes, okay. You can. So I can go to something else, no? Yes, I think I will be on scene also to them. So
okay, thank you.
It's okay. Yes.
So one student, Divyandu Das, he has two questions. So first question is, targeting an enzyme may lead to impairment of normal cellular machinery. So how to deal with this while we are designing a drug for a particular purpose?
Yes, very much. I'm so thankful that my talk, at least some part of knowledge has been, actually seep in at least to some of them. I really never heard this question most often when I give talks in the classes, etc. Everybody consumes it, but nobody thinks about it. I'm thankful to you, the question a person who has done. Actually, this is a balance. Every drug will have a bad effect. Not only it will cause imbalance in that pathway, it may cause imbalance in many pathways as well. And that is the reason kinases, anti-kinase or inhibitor of kinase to become drug becomes havoc problem. Very few are really been successful. And mostly in cancer, the reason is the cancer patient doesn't have much choice. It is a very interesting question. Always you take a balance out of it. Hence, that's why target selection becomes most important. The least amount, the most important blockbusters are picomolar amount inhibitor. In KIs are almost picomolars, both for penicillin and for omeprazole. So I had worked with omeprazole's next level short-acting inhibitor of omeprazole, but it was a very difficult for AstraZeneca to put that compound in because omeprazole was blockbuster. And the reason was the very low amount affects the pathway, which was HK-ATPase inhibitor. Actually, it was binding to HK-ATPase as a covalent bounder. So not non-competitive, not competitive, but covalent. Hence, even penicillin does the same. So you can see this, that as lowest possible concentration the enzyme is inhibited is the key for success. So that's why you have to select proper target. Okay. Next question. Okay.
Yeah, next question. Yeah, Divyantu Das's next question is, how does the molecular evolution of enzyme target interface with the effectiveness of a drug?
The enzymes do have a choice to evolve, and many does. Virus, of course, does. Viruses are much faster changing their enzymatic characteristics. And to tell you the truth, the best work has been done by DHFR and TS modeling long back in UCSF, actually. Many molecules were designed and tried to be inhibiting DHFR, and becomes this series of drug was for anti-malarial and pyrimethamine. Whole series of drugs were built and found to be immediately resistant within 5 years. The reason is the probability is that some enzymes do evolve faster and change. Many enzymes do change their active sites here and there, but if they become ineffective to the inhibitors or the drugs, it is really a greatest failure. HIV is another case. Huge number of strain of HIV across the board has come up, and presently we are having trouble with Mycobacterium because Mycotuberculosis bacteria has tremendous resistance. Malaria also has tremendous resistance, and this resistance are due to their changes in their cellular mechanism to many ways, many methods by which the pathogens try to throw away drug. One of them is throw away drug. One of them is to so you have to work with transporters. Then one of them is to just change the enzyme active sites. Hence, this is a fight is for a long time with our designing qualities and bacteria's evolution. So we cannot, even already we noted in many strains in India, my friend Partho, they are doing a whole genome study at NDGMS Kalyani, and he told that many of those are already, the strains in India is probably would be resistance to some of the spike invaders or spike busters or spike inhibitors. We don't know because many places the spike proteins have changed. So yes, once more you are talking about a balance, how fast we can make a drug and how faster they survive by changing. So we try to do that. However, here comes the trick of you may say the trick of drug designing. What we have seen that if it is uncompetitive inhibitors, that is, it goes to the transition state analog designing. Designing is very tough. Even assays are very difficult to design. However, if we make that, it is unlikely the proteins can be changed because the information what goes back is from competitive inhibition, that these are the places they are blocking. And that information somehow feedback and evolving of proteins happens. However, if you do the transition state, doubtfully that it may not be surviving so much. Number one. Number two is covalent binder. Designing of covalent binder, two successful answers are aspirin and penicillin and your case of omeprazole. Both are covalent binder, and none of them got anything to do with the only thing they changed, that penicillin really got changed, that it started making an enzyme which will cut the penicillin beta-lactamase. So, it improvises to cut its own leg or something. So it improvises that. Hence, we had to go for cephalosporin and other systems which will stop that as well. So this fight is always there. Always there. Okay. Any more? Hopefully you get the answers.
Yes, ma'am. Thank you, ma'am. And another question. Yeah. there are many proteins involved in multiple pathways. There are many proteins involved in multiple pathways. For example, in one particular pathway, a pathway gets dysregulated. Will it be feasible to target that protein?
It only depends on how many associated pathway supports it. Very much you have to look at associative pathway. I always suggest my students also, when they are designing molecules to go for targets, you have to observe what are other metabolites are working in the pathway and what are their cognate, that is their corresponding other partners. That is, these metabolites may not be functioning only on that pathway, maybe in other places. As I shown you one caricature or one thing, and many examples I could show, but it's a bit clumsy. You can get another metabolite replaced from other part or modified from other part. In that case, your selection will not be good. Hence, you must see the cognate pathways, not only the pathway which you are interested, but other pathways. Okay.
Thank you, ma'am. So
Yes.
one last question I would like to take, ma'am.
Okay.
How can we calculate inhibition of irreversible inhibitor?
Irreversible, you can actually, by simulation, you can do everything, to tell you truth. But biologically, how you do that is a bit of a problem, because most of the reactions are reversible, except few reactions seem to be irreversible. But so one needs to be, experimentally finding will be really tough. However, what one can do is, you can design compounds which will be irreversibly binding and test the assay. Test it in assay, whether the free molecules, free ligand, how much you can assay. Receptors case, people do that kind of assay. In receptor designing, people do calculate not the binding receptor, but how much freely available. How much available is freely, that the antagonist or agonist. So one can design that kind of experiment for your purpose if you want to find which is called irreversible reactions. Basically, the penicillin binds to the pathway of where one enzyme is there, which it binds, penicillin binds, because it mimics the substrate actually, not the product, but the substrate itself. And it goes to the ES step and just binds there. And it forms a covalent bond. Not only it binds, it forms a covalent bond. And so penicillin became so successful in the sense. Omeprazole, on the other hand, it also forms a covalent bond, but it has a very different mechanism. Actually, it's very interesting. Only at lower pH, which is in the stomach when you have acid circulation is very huge, and that is the cause of having the problem, then it goes and forms the disulfide bridge. However, with the HKT base, which is available in the stomach. But the moment it becomes neutral, still it stays, but it becomes a little bit higher than neutral, that is on the basic side, it releases off. It doesn't stay. And that's how very lowest possible inhibition or lowest possible amount of molecule of omeprazole works so well. So that is also a mechanism by which it's a not a reversible mechanism, it's a irreversible mechanism. However, this kind of enzymes, you have to really go through and plan it. We did find one like that in our paper in anti-malarial. We have highlighted that probably we should target that molecule against that particular enzyme. It was in non-mevalonate pathway, one of those. It's there in my reference, the paper is there, a few years later, paper we published. However, not many people design that kind of compound, so it's a bit tough to design compounds. Once upon a time, the molecules used to be built according to you require. Nowadays, molecules are there. People just take it and just try and throw. Nobody, sadly enough, synthetic and medicinal chemistry is becoming very backseated. Then we will never explore new molecules. And most of these new targets probably will be ready by a lot of ways. However, maybe the compounds may not be available in that case. And so it's becoming a topsy-turvy in some other way, I would say. Yes.
Thank you, ma'am.
Any more questions? I have exhausted too much time. I think 1.5 hours. No, not much, because you said 1.5 hours for the question answer. Okay.
So,
Okay.
Participants can view your video later on also. It will be in MHRD YouTube channel, and they can write in the questions in the comment box, and also they can write to us, and we can also we can also put their questions to you. So there will be.
Please.
Yes, ma'am. So thank you very much, ma'am, for explaining the questions, and we are really happy to have you here. So on behalf of DDH training team chairman, Dr. G. Narahari Sastry, we thank you so much for our invitation and sharing our thoughts and ideas and giving a wonderful lecture here. You have started with the basics of enzyme kinetics reactions, then its applicability in vitro experiments, then target identification in pathway, target strategies, and few case studies you have shown, and many more. So it is a very wonderful session. It is our privilege to hear from you, ma'am. So thank you so much for being with us.
Thank you for giving me the opportunity, and it will be successful if the students or participants do take time, go through it and send me questions.
Yes,
maybe many errors may be there or something. If they find, I will be very happy to improve. I had this whole my talks in the you would see in my CV that it is online in the under the NII or some. Now they have put it on YouTube apparently. So many questions I get even after, those have been done in 2018.
Yes.
And I get questions this year, many students, and actually many of them said that we don't have any class. So can we access your access and study through all those? I said, yes, go ahead. So I'll be very happy. And once you make this YouTube, I will be happy and I can circulate to other people. Whoever is interested to go through. And that's
definitely,
the beauty of
we'll send you the link to your mail.
Link. Just send me that. That will be fantastic. And send questions even after 10 days or 15 days.
Yes, after maybe 10-15 days, your viewers will be increased maybe 2,000, 3,000 to 4,000.
No,
so
I'm sure
getting many questions. I hope so.
No, it's not the number matters. It matters how serious they are. Actually, the first person what you said, he was really listening and asked very pertinent questions. So there will be very few people who will be interested on these topics. But I am purely optimistic. This will be the drug design for next future generation. That's why I showed you two types of drug design. One just repurposing. Another is really new novel interaction. And that's the most important. Okay.
Thank you, ma'am. So we will wind up the session here. And actually, there is little technical problem now. From now, the YouTube stream will not be there, ma'am, because it's 5:30. So
no, you will put it later, is it?
Yeah, it will be there, ma'am. It has stopped.
Okay.
Now it has stopped. Yeah.
Thank you very much. 5:30 is over, so we should close it down.
Yes. Thank you so much, ma'am.
Okay. Good night to Hriday and you both.
Yes,
okay?
He's here, ma'am.
Thank you.
Good night, ma'am.
Good night, ma'am. Oh my God. मेरा दिमाग.
What will happen? These